Welcome all. Thank you for joining us this morning with the Science Advisory Committee. We are here with Dr. Koty Sharp from Roger Williams University, who is going to talk to us all about microplastics and vectors for disease in corals in Rhode Island. So I'm going to let Koty take it away.
Great. Can you see my slides and can you hear me?
Yes, everything looks great.
Perfect. Okay. Well, thanks so much for giving me the chance to speak this morning. I was sad to miss the event back in October when I know many people gathered to talk about the urgency of microplastics pollution across the globe, but also especially in Narragansett Bay. And my specialty, my background is in microbiology and really in the microbial ecology of animals in marine ecosystems. And I got into research on microplastics from that perspective. So rather than sort of quantifying plastics in the environment and looking at how many plastics are interacting with animals or other parts of the food chain, what I really have been sort of fascinated with and frankly concerned about is the part of microplastics pollution that's really hard to see, the microbial aspect. What is the impact of plastics pollution on the microbial members of marine ecosystems, and how do those microplastic pollutants transform or have the potential to change the way that animals interact with microbes? Most importantly, are there different microbes that are in the marine ecosystem that are now novel and being introduced to animals in the marine ecosystem? And so that's been driving a lot of the research that I've been doing over the past few years with students in my laboratory here at Roger Williams, and in addition to collaborators across the New England area. And so the work that I'll be talking about today, is really based on that. And I think to start off, it's really important to acknowledge that this is work that is inspired by Linda Amaral-Zettler and Eric Zettler and others who explored the microbial ecology of plastics pollution a decade ago, as they started to sort of ring the warning bells that marine microplastic pollution is enriching for and creating unique microhabitats for novel microbes. And so we have tons of particles, of course, in the marine ecosystem, silt and other naturally occurring particles. And those particles enrich for particular bacteria that are involved in biogeochemical cycling in seawater. And they deliver and often are vectors for geologically and biochemically significant processes in animals and other portions of the ecosystem. But what is of a significant concern is that plastic particles enrich for what has become known as the plastosphere, biofilms of microbes that typically exist either in low abundance or in such low abundance that they're really hard to detect in the marine environment. And microplastics associated biofilms are highly variable. According to surveys that the Zettlers and others, especially from Woods Hole Institution, have run across the globe, it's really clear that that the plastosphere is distinct from biofilms that occur on naturally occurring particles in the marine environment. And so with that context, what I've sort of been thinking about is what's the impact of having the sort of enrichment of novel microbes in the marine environment? And of course, I come to this from an animal microbiome perspective. We all know at this point the importance of microbiomes for animal health, right? And I tend to focus not as much on pathogenic microbes, but on beneficial microbes. We know that microbiomes are very important for animals in terms of digestion, especially for humans, right? But microbiomes also are extremely important. They play a role in controlling the development, the early development, the immunity, protection against pathogens, and other, other key processes in animal health. We know that that there are certain syndromes and disorders and diseases, including everything from um, uh, diabetes and neurological disorders that are actually impacted by animal microbiomes. And so the animal microbiome plays a major role in health and wellness. And so as we're thinking about that and we're thinking about animals being presented with novel microbes via microplastics pollution, the concern really is, how do microplastics pollution impact animals? Can they alter animal microbiomes? And what's the impact on animal health? So the organism that I work with in my lab is the northern star coral, Estrangia poculata. We found this to be an extremely useful experimental organism for a variety of questions in animal microbe symbiosis, especially considering this in the face of climate change and in the face of pollution. It's an extremely robust organism in it's can withstand experimental manipulation and a lot of handling in the laboratory. It is, unlike its tropical coral cousins, it's not threatened. And it's highly abundant here in Rhode Island waters. We use it as a model system for understanding a lot of different aspects of symbiosis, and really started this work based on the fact that it engages in a flexible symbiosis with algae that live inside of its tissues. So unlike tropical corals, which require those algae to live, Estrangia can exist either as the way you're seeing it here in this picture, brown, sort of full of those symbiotic algae, but then also in white colonies where it has very, very low concentrations. And that allows us to tease apart the influence of those algal symbionts on other processes in coral physiology. For example, being able to withstand thermal stress and disease and other environmental disturbances. So for the sake of microplastics, the character of Estrangia that's extremely important is the fact that it is an aggressive feeder. So unlike tropical corals, it takes in a lot of particles from the surrounding water. And that becomes really useful in laboratory assays as we're trying to understand the dynamics of ingestion of plastics particles. And so here's an example of what we're looking at here is zooming in on a single animal, and this is the mouth. This entire animal here is what we call a polyp, and so this is the mouth of the coral here, and then this is a ring of tentacles that are extremely long and used for prey capture in the seawater, and then the blue dots that you're seeing are polyethylene microbeads that we presented to it in a laboratory assay. And then if we take a look at the video, you can see that the beads, those microspheres that are about 200 microns in diameter, are taken right in. And so this is something that I mainly show this video just to show that it's really easy to see on microscopes. It's easy to monitor ingestion and, in fact, egestion, or beads that are then rejected from the coral and sent back out, especially because this coral has relatively clear tissue. It's easy to work with in the laboratory. So what I wanted to do is just sort of run through some of the main findings that we have made so far using Estrangia. The first one that I want to point out, and this was work that I did with Randy Rojan from Boston University and Juanita Urban-Rich from UMass. The first thing that we found that was really quite shocking was that there were on average, over 100 particles of microplastics per mouth in a coral colony. And so I want to go back just for a second to show you this picture. So in this picture, this is a single Estrangia colony. Each one of these little circles here is a single polyp containing a single mouth. And so the counts that we did suggested that per polyp, per mouth, there were over 100 pieces of microplastic in each one of those polyps. And these were from populations, from colonies that we collected in Fort Wetherill in Jamestown, Rhode Island. So right here in Narragansett Bay area. And we classified, most of them were fibers, some of them were beads, and then other small miscellaneous shapes, but most of them were the fibrous shape. The next thing that we found was when we took Estrangia colonies back into the laboratory, what we found was that if they were given 50-50 choice, so an equal concentration of polyethylene microbeads, which you can see on the right-hand side in this picture that are sort of the bright blue, and they're giving a 50-50 choice of polyethylene microbeads or of brine shrimp eggs, which are approximately the same size and same shape. Overwhelmingly, they took in higher quantities of microplastic beads. And so what you're looking at on the left is a graph that's showing the amount of microplastics per unit volume of the coral versus the amount of brine shrimp egg ingestion per unit volume. If it were completely random or 50-50, reflecting the concentration of the beads versus the brine shrimp eggs, what you would see is a distribution of these points here reflecting this one-to-one line. But instead, there was a leaning toward the microplastics. And in fact, this was statistically significant over a dozen trials where microplastic beads were presented to the corals in equal concentration to the brine shrimp eggs. So they take up more polyethylene than actual food when given the choice. And I think the other thing that's really important to visualize, I think this is the easiest way to show this. Now, this is a sped up video that my lab technician took. When we give the corals, now granted, this is in a laboratory assay with a really, really high concentration of beads. But what I hope you're able to see here is that the coral is taking in these beads through the mouth. There's tentacular movement, and especially if you look across the rest of the coral colony, you can see that there's movement of tentacles that are sort of handling the beads. One of the things that happened before we started taking this video is that a lot of the colony sort of moved and puffed to move some of the beads away from the mouth, away from the oral disc. Overall, there's energy that's expended in this non-passive interaction with microplastics. So when you can think about it, if you have something on your body that sort of comes in, you know, it's not something that you want, you dust yourself off, right? That takes energy to do that. Now, of course, wiping your arm off isn't going to influence the amount of energy you have left to do other things like go for a run or eat a meal. But it's a substantial amount of energy expenditure. At least this is what we were hypothesizing as we were watching corals process plastics in the laboratory. And so these non-passive interactions with foreign particles that, and by the way, the other thing you're seeing in this video is that they're taking them in, ingesting these particles, but of course they're not digesting them for any sort of nutritive property. And in the video, you can actually, if you take a look, it just finished doing what we call egesting, right, or getting rid of these particles. And so there's energy taken or expended to take all of these particles in, and then energy expended to egest those particles. There go a couple beads now. And so the question that we had is that energy substantial enough to influence subsequent activity? So if there are particles that are presented to corals, and then and the corals are then processing the particles, does that influence their ability to do other important activities such as eat something and acquire nutrition? Oops. There we go. And so what we found was when we presented the corals with microplastics and we incubated them with microplastics, it changed their subsequent feeding behavior. And so the graph that I'm showing you here is on the top. This is measuring the amount of copepods that were consumed in two different trials. And so in the black, this is representing the amount of copepods that were consumed after brine shrimp eggs had been presented to the corals. And in gray, you're seeing about the same amount of copepods were consumed after they had been exposed only to microplastics beads of the same concentration. And then we did this again with live brine shrimp, another food source that Astrangia is really great at eating. And then we did it with the brine shrimp eggs. So these are these small little spheres that are about the same size same shape and same size class as those microbeads. And so after brine shrimp eggs, they were then presented with brine shrimp eggs and they ate a certain amount. After they had been incubated with microplastics, we saw no uptake of those brine shrimp eggs. And so this led us to believe that there are energetic consequences that are due to the ingestion, the handling, the exposure of microplastics in some way the microplastics exposure changes their subsequent feeding patterns. And it was interesting that that only happened in the food that we offered them that was similar in size class and in shape to the microbeads. So then another thing that we have been able to determine in the lab using advanced microscopy with fluorescently labeled bacteria is that microbeads, and in this case, again, these are these small polyethylene microbeads, are able to deliver novel bacteria into apoculata as well as onto the surface of apoculata. So here I have a couple of students here at Roger Williams who are using the confocal laser scanning microscope to localize bacteria inside of the inside of Astrangia. And what you're seeing here is microbeads that are UV fluorescent. But then on top of the microbeads are small E. coli cells that are labeled with green fluorescent proteins. We call these GFP E. coli. And GFP E. coli cells, when we grow them up in culture and then we incubate the microplastic beads in those cultures, they'll form a biofilm on those beads. And so E. coli is not sort of the main pathogen of concern out in the marine environment, but as far as a laboratory assay, what we were able to test was whether or not E. coli that's in a biofilm on a microplastic bead and then presented to Astrangia, do we see that E. coli inside of Astrangia as a result of ingestion of the microbeads? And indeed we do. So what you're looking at here is a polyp of Astrangia under a dissecting microscope with fluorescence. And so this entire piece here is the polyp of Astrangia, and you can see the tentacles here. The blue glow is from a group of beads that are inside that have been ingested into the mouth, and then the green glow that you're seeing is from those green GFP tagged E. coli that have basically ridden in on those beads into the gut cavity of the coral. So can they be taken in? Yes. And then one of the main questions is what's the fate of those bacteria after those, you saw in the video, those beads get egested. And we're still working on that, but we have some imaging now that suggests that those E. coli basically are dropped off and left behind in the gastric cavity, as well as on the surface of apoculata. Another piece of the finding that was published in this same paper was that after corals had been treated with microplastics coated with E. coli, 9 times out of 10 those corals were exhibited mortality. They died from that treatment as opposed to microplastics alone. And so microplastics in the presence of this, this bacterium was able to actually elicit lethal results on the coral. The other piece of this story is it possible for, or I guess the other question I should say that we've been asking is it possible for this coral to take up beads indirectly via their prey? So copepods are another great prey source that we use, or food source that we use in the laboratory, and in the wet lab we feed Astrangia with these copepods. When we incubated copepods with microbeads, as you can see here in this left-hand panel, the copepods would end up having these beads either on their surface or in some cases even inside of their gut cavity. And then when we fed those copepods to Astrangia, we could see, so what this is actually looking into the inside of the gut of the gastric cavity, I should say, of a coral, and we can see the outline here of a copepod that has been taken up and that has beads inside of it. And those beads are now inside of the Astrangia gastrovascular cavity. And so we know that they can take up beads either through their prey, they can take up beads directly, and we're still working on sort of making the links of what happens to the bacteria that are naturally occurring on those beads that then are in the prey that are then ingested by the coral itself. And so as we're sort of uncovering and starting to visualize and image these processes more, we're really sort of uncovering other questions that we think are important to continue to address. And so we, like I said, we know that they ingest and they interact with microplastics. They can take them up directly. They certainly seem to be able to take them up indirectly via their prey. We have an indication that there's energetic consequences to their handling. And I should mention here that we're not the only people who have found that. That's now quite consistent with a lot of other groups from around the globe who are finding evidence that there are energetic and ultimately fitness consequences just from handling plastics. And then most importantly, we know that microbes can basically hitch a ride and go into and onto the surface of animals via microplastics. And so with those questions in mind, one of the things that we're now focusing on is trying to understand how the delivery of microbes from microplastics to animals impacts the microbiome and ultimately the fitness of animals. And so very quickly, what I want to just share with you is some of the other work that I've been doing in collaboration with some folks on tropical corals. And tropical corals, of course, have been receiving a lot of attention because of massive global outbreaks of coral disease and something called coral bleaching, which is the loss of algae from the tissues. And as I said earlier, those algae are required for the survival of corals. Here is a group of corals that are showing the algae in this sort of dark brown color, and then corals that have been thermally stressed from human-induced ocean warming, and now are what we call bleached, or basically they've lost the algae that are in their tissues, and if they don't reacquire those algae, they ultimately basically starve to death, and then the corals die and become a beautiful skeleton ground for algal overgrowth. And so we're seeing massive habitat loss and destruction as a result of ocean warming, disruption of this symbiosis, and coral bleaching and coral disease. And the reason that I bring this up as we talk about microplastics pollution in the marine environment is that it's been pretty clearly shown that microplastics and the occurrence of microplastics I should say, are correlated with increase in coral diseases. And so where we have regions of very, very high microplastics pollution around coral reef regions, we also see an increase in coral disease. And this picture was a photo that I took from an article in The Atlantic about some recent work by Dr. Jolie Lamb on linking microplastics pollution to coral disease. We don't totally understand how that could happen, but the idea, actually I'll go back to this just for a second, is that if you take a look in this picture, you can see, so here's the Nike swoosh symbol of something that has made it onto a coral colony. And these are sort of finger-like pieces of a coral colony that have likely been sort of rubbed or in some way a physical contact and physical disturbance can cause lesions that then leave the coral vulnerable to infection and or disease. So you can think about if you have a cut on your hand or on your arm, and then you go swimming in the water, that could potentially become infected. One of the problems, of course, with climate change is that as temperatures in the ocean increase, the microbial landscape across the board is changing. And there's been a lot of work done on this recently showing increases in pathogens and at the very least changes in the microbial communities in seawater. And so as ocean warming is compounded with increases in these physical insults, right, this microplastic rubbing on corals, it's likely that the consequence is that corals are now susceptible because they've been given some kind of mechanical injury or lesion, and they are susceptible to infection by novel pathogens that are now in increased concentrations in the water because of climate change. And so some work that I've been doing with Colleen Bove out of Sarah Davies' lab and Nikki Fogarty at UNC Wilmington and her graduate student Charlotte Sujerski has been focused on the staghorn coral, which is a threatened coral from the Florida Keys. Very briefly, I'll just tell you that what we did was we exposed fragments of this coral to ambient temperatures, ambient pH, and then also predicted temperatures to represent ocean acidification and warming. So higher temperatures, lower pH, and then with and without microplastics fragments. And we were really interested in understanding more about whether or not microplastics compounded with ocean acidification and warming can change the microbial communities of corals and whether or not that has fitness consequences on the corals. And this is work that was is in review right now, actually in revisions right now, and should be released to publication in the next week or so. And I'll just tell you very quickly that the punchline was not at all what we expected to see. So all of these different stresses together or even separately caused a massive immune response by the coral. So there was clearly some kind of indication of an injury or an insult to the coral, but the bacterial community did not change as a result of any of these different treatments. And so if you take a look at this figure, what this figure is displaying is basically just a control where there's no environmental stressors added to the coral. This is ambient temperature plus microplastics alone. This is ocean acidification and warming with no plastics. And then this is ocean acidification warming with microplastics. And in all cases, what these different bar graphs are showing is basically the proportional abundance of different microbes in the tissues. And in every single one, they're all dominated by a single microbial type. And there seems to be very little, even when we remove that one single dominant group of bacteria, there's no variation from one treatment to the next. And so, although there's a massive upregulation of immune response on the genetic level, or gene expression level, I should say, in these corals, we don't see a change in what kinds of bacteria are in the coral. And so now we have a whole slew of new questions that we're trying to ask. So is this, and this bacterium that's represented in the green is really a regular bacterial associate that's been shown to be present in a lot of corals across the Caribbean. And so the question is this bacterium itself changing from friend to foe? Is it changing its behavior and becoming more pathogenic? Which is something we wouldn't be able to capture in this kind of analysis where we're just saying who's in there. And then one thing we didn't look for in these samples that in hindsight would be a really interesting thing to ask is this changing the dynamics of what viruses are in the tissues? We know that viruses are very common in the marine environment, in animal tissues. Many viruses are lying latent and are able to cause harm to animal cells and to other bacterial cells. Others, in fact, are completely commensal and have no impact on the animal fitness. But with changing temperatures, with changing acidification, with changing microplastics, we don't know the impact on what we call the virome, on viral abundance or viral behavior. And it could also just be something else that we haven't come up with yet. But overall, what we're able to see is that plastics and warming cause a change in the immune response. And by the way, none of these corals none of these corals died in response to these treatments. And so all of the responses that we're trying to measure are what we call sublethal responses, right? So they definitely exhibited a massive shift in the expression of immune response genes, but we don't really know what was causing that. And so could it just be the mere presence of plastics, the changes in thermal stress? And that's something that we're definitely interested in looking at more. But overall, I think what we do know is that microplastics alone are not always a major threat to animals. They may not kill them, but they do pose a substantial threat, especially in combination with other elements of global change. And I would say that it's really important for research to focus on synergistic interactions because, of course, microplastics pollution isn't existing in a vacuum, right? It, of course, is happening at a time where we have massive problems due to climate change, including changes in the microbial community in the seawater, increased levels of pollutants, which I know were discussed a lot at the Plastics Forum in October. And it's really important to sort of focus our research, I think, on these sort of harder to see and harder to detect sublethal impacts and microbial aspects of microplastics pollution, especially in the context of climate change, ocean warming, and acidification and increased pollutant levels, as we're trying to construct models to understand how microplastics impact coastal ecosystems. And so with that, I've taken a lot of your time, but I just wanted to acknowledge funding sources. I've had a lot of students working on these projects over the years, and especially collaborators Randy Rogen, Nikki Fogarty, and Sarah Davies. And if there's time, I'd be happy to take any questions.
All right, all. So yes, if there's any questions, you can use the raise hand function or pop them in the chat. While everyone's thinking, I found it really interesting that the brine shrimp eggs, when you fed them to the corals and then fed them microplastics again, they actually they would take the brine shrimp eggs over the microplastics in that thing. And I thought that was super interesting because that's not contradictory to what you showed before, but it absolutely shows that they're recognizing that maybe microplastic beads are not a food source that they can get energy from. And I was wondering how important brine shrimp eggs are, since they're in the lab and it's great to see, but in the wild, like how important are those relative to the copepods or larger brine, or, not eggs of brine shrimp, but actual brine shrimp?
Yeah, so I think there are a lot of questions in there. I think the first point is that we're actually still not sure what Astrangia really eats in the wild. It's interesting. There's been a lot of work done on that, and early on there was a work examining the gastrovascular cavity contents of Astrangia from wild collected samples, and mostly what we think they ingest is sort of particulate matter from the water column. But we do know that under a lunar cycle, there's this very specific feeding behavior that happens where there's an amphipod that actually will sort of crawl into the mouth of the polyp. And so we find amphipods according to a lunar cycle in the polyps. We know that in the laboratory, their food preferences are extraordinarily flexible. And I will even say that there's some anecdotal long told stories from researchers at URI that even hamburger meat could be taken up by Astrangia. But in all seriousness, brine shrimp eggs, copepods of several different species, and brine shrimp in the sort of juvenile forms are all taken up readily by Astrangia in the lab. So the fact that they were not taken up at all after that microplastics exposure was really striking. And I don't know that it indicates so much that they show a preference, and what I didn't show is that we've done a lot of pairwise comparisons where brine shrimp eggs over juvenile Artemia and other, they'll take them all up. There's absolutely no preference exhibited. So I don't know that it's a preference so much as, but I do wonder about this, size class or shape or something else that has to do with, why the ones that are so similar to microplastics. I don't know.
Oh, it's interesting. Any other questions before Koty runs to her class? All right. Thank you so much for the morning, Koty. We look forward to working with you on lots of this, and so excited you could share this research with us, and everyone can look for it when we update our website and get all these videos on. So thank you kindly, and we will chat soon.
Okay, thanks so much for having me. It was great to be here. Bye-bye.